Welcome to Lecto. Today we are going to discuss viral growth cycle. So in this cycle you will see the three most important event: early event, middle event and the late event. In the early event you will see the three most important phases. First one is the attachment, second one is the penetration and third one is the uncoating of the capsid that is surrounded by the viruses. In the middle, you will see the mRNA synthesis, protein processes, and the genome replication. And in the last phase, you will see the assembly of the virion, and you will see the release of the virion from the host cell. So these are basically the different events. But in this lecture, we will just discuss the early events. In the early events, we will discuss or we will cover the attachment of the virus to the host cell, that how virus actually attach to the host cell. Second one is the how viruses penetrate into the host cell, and last is the uncoating of the viruses, because as we know that the viruses are actually surrounded by the capsid, and capsid is made up of protein. So actually breakdown of the capsid is actually called the uncoating. So first of all, if we discuss the attachment, that how different viruses attach to the host cells differently. So if you see here, like this is the host cell, this is the host cell. Okay, now this is the virion. Okay, if you see here, host cells has a receptor. So there is a receptor that is actually present at the surface of the host cell. Okay, and the virion has different proteins. Okay, now you see there is a protein that is actually present to the virion, or these proteins actually act as a ligand. So these proteins will bind to the specific receptor, just like the enzyme substrate complex. Specific enzymes will attach to the specific substrate. Similarly, specific proteins that act as a ligand will attach to the specific receptor. Okay, it's mean that there is a specificity between the cell and the virion. So actually you will see the specificity at the organism level and at the organ level. So it means not all viruses affect all organisms. Okay, this is the case one. And not all viruses attack the all organs of the body. So this is another case. If we discuss like poliovirus, poliovirus infects or enter the cells of the humans and the other primates, but the rabies virus can enter all the mammalian cells. This is a most important point, that the poliovirus and the rabies. So actually there is a difference. Primates is actually the order of the mammal. Okay, poliovirus can infect the specifically only the primates, including the human, but the rabies will infect all the mammalian cell. This is the most important point. Actually mammal is a broad range and the primates is a narrow range. Okay, if we discuss organs like specific parts of the body, then you will see the HIV. So HIV basically affect most importantly the helper T cell, because helper T cells have receptor for the attachment of the HIV, human immunodeficiency virus. So this is most important thing. So it's mean that a virus can infect only specific organism, and in that organism you will see that viral will attach to the specific parts of the body as well, because that part has a receptor. So this is the thing. If you see here, like this is the helper T cell. Okay, And this helper T cell has a receptor, and that is called the CD4 receptor. Okay, this receptor function normally, as you see that this receptor will bind to the major histocompatibility complex type 2. So if you see here, like this is the major histocompatibility type 2. Normally this complex will attach to the CD4 receptor and activate the helper T cell. So actually major histocompatibility complex actually present at the antigen presenting cell. So antigen presenting cell having major histocompatibility complex 2 that will bind to the CD4 receptor and activate the helper T cell. This is the normal function. But if you see, if I say that when the HIV enter in the body and this virus has ability to attach to this receptor as well, CD4 receptor, and these viruses having ligand that has ability to bind to this, and that will infect the helper T cells in this case. So it's mean that the receptors are normally present and that they function properly or normally. But when the virus entered, that virus has ability to attach to that receptor as well. If you see here, different example like rabies virus. Rabies virus has ability to attach to the acetylcholine receptor. So if you see here, rabies virus has ability to attach to the acetylcholine receptor. Okay, that is actually present at the level of the neuron. Okay, Epstein-Barr virus has ability to attach to the complement receptor. Complement receptor. Okay, you see that the specific virus attached to the specific part of the body. Herpes simplex type 1 virus has ability to attach to the fibroblast growth factor receptor. Fibroblast growth factor receptor. Okay, and the vaccinia has ability to attach to the epidermal growth factor receptor. Epidermal growth factor receptor. So here are basically the different situation that the specific virus attached to the specific part of the body. So this is the attachment. There is another important thing. If you see, virus can attach to the host cells by the fusion as well. So what actually the meaning of the fusion? So if you see here, we discussed this is the host cell, and here are basically the two viruses. First, if you see, this is actually the influenza virus, and second is the coronavirus. Okay, these are basically the two viruses. In the influenza virus, you will see, if you see, this is the reddish area. This is the heme agglutinin. Okay, but in the coronavirus, you will see the spikes, protein spikes. In both situations, you will see the presence of the protein, and that is the fusion protein. Fusion protein actually present in both viruses, influenza and corona as well. Okay. Heme agglutinin, as the name indicate that it's mean there is agglutinations of the RBCs. Yes. Heme agglutinin are basically the glycoproteins that are actually attached to the RBCs and agglutes the RBCs. So, whenever you see that these heme agglutinin and these spikes, protein spikes, actually mask the fusion protein, and fusion protein is very much important with the attachment or the fusion of the host cell. So, in that situation, you will see, first of all, we have to remove the heme agglutinin from the influenza virus and the spikes protein from the coronavirus. So in this case, you see lot of different protease is actually present in the plasma membrane. Enzymes can also present in the plasma membrane as well. So in this case, protease is actually present in the plasma membrane. These proteases actually you see they dissolve or destroy the spikes proteins and the heme agglutinin. After its destruction, you see when you see these spikes proteins and the heme agglutinin removed, then you will see the reveal of the fusion protein. When the fusion protein reveal, then these fusion protein will attach to the host cell, and in this case you will see the fusion of the viruses to the host cell, and in that situation these viruses can easily penetrate into the host cell. So this is another important thing. So when the virus enter into the host cell, then what will happen? So in this case you see like this is a virus, okay, and that virus has a genome in it, okay. When this virus enter into the host cell, basically this is the host cell. When this virus enter into the host cell, it will enter you see like this virus enter into the host cell, and this virus will carry the plasma membrane from the host cell. Now this virus surrounded by the plasma membrane of the host cell is actually called the vesicle. This is the vesicle, okay. Now it will move further. In the vesicle you will see the pH is low, okay, and due to the low pH in this vesicle you will see the pH is low, and due to the low pH you will see the fragments of the capsid, or you can say capsid will break down into different fragments due to the low pH. Now in this case you will see the capsid is broken down into different fragments and the genome is ready or free from the capsid. So this breakage of the capsid is actually called the uncoating process of the viruses, okay. Now this genome can exit from the vesicle, okay. It has two pathways. First is the vesicle membranes can break down, okay. When the vesicle membrane break down, then you will see the genome will enter into the cytoplasm, okay. There is another process that this genome can exit by itself into the cytoplasm without any breakage of the vesicle membrane. So here are basically the two processes. First one, the genome can enter into the cytoplasm with the breakage of the vesicle membrane, and that genome can enter into the cytoplasm without any breakage of the vesicle membrane. So these are basically the two processes through which the genome from the viruses can enter into the host cytoplasm, okay. And last most important thing is the nucleic acid. Nucleic acid. So in this case you have to remember this sentence that all viruses are infectious, but not all purified genome, either DNA or RNA, is infectious, okay. If we discuss this, then you can easily understand what actually this sentence say. So if you see this is the cell of the primates and this is the cells of the non-primates, okay. We discussed that the poliovirus infects the primates only, okay. If you see here, like this is the polio virus. This is a virus having capsid outside and genome inside. And this poliovirus, in the second case, you will see only the genome of the poliovirus. Now, this poliovirus will infect only the primates. Why? Because this poliovirus has a protein, and that protein has ability to attach to the specific receptor that is only present to the primates. So poliovirus will infect only primates, and this poliovirus is actually surrounded by the capsid, and capsid has a protein. So this polyvirus will attach to the primates only, and this polyvirus has not ability to attach to the non-primates, because these non-primates has no any receptor that is compatible with the ligands of the polyvirus. This is the case one. When I remove just the genome of this poliovirus, now this genome doesn't require any receptor for the attachment and the entry of the cell. Now this genome has ability to enter into the both primates and the non-primate cells. This is the case. I'm saying that the all genome has ability to enter any cell without any specificity, but the virus having capsid will only attach to the specific host cell. This is the most important thing. If you see, like after its virus replication into the host cell, you will see the different copies of the viruses in this case. Okay, but there is another thing. But in case of genome, when the genome enter into the non-primates, then it means after its replication into the non-primates, then you will see the different copies of the viruses. Okay, but now this time the productions of the virus, in this case you will see the lot of capsid that is actually surrounded by the genome. This is actually the normal polyvirus. Now this normal polyvirus cannot again infect the non-primates. It will only infect the primates only, because now this virus has a capsid, and capsid has ligand, and ligand is specific to the attachment of the specific receptor. So this is the case. So that's why all viruses are infectious. So this is the case one. But viral genome is not always infectious. Why? Because in some times you will see the genome of the virus will integrate with the host cell DNA or genome, but it will not cause any disease, and it will goes to the progeny cell without any hazards. So this is actually the different phase. You see the lytic phase and the lysogenic phase, do different phases of the viruses. So this is the normal process. So this is the case. So this is all about the viral growth cycle. If you have any question, you may ask in the comment section. Thank you so much.